Our next presentation is Dr. Andrew Baird from Health Sciences and the Surgery Department on avatars of human disease, personalized modeling for personalized medicine. Very exciting.
Thanks. I'll try not to let you down. Thank you for waking everybody up, by the way. Don't start the clock yet. Thank you for the opportunity to share with you a vision for where UCSD might think of going in its strategic planning for human health and society. I really represent a team. We're actually a loosely grouped organization. We're informal, we're grassroots, and we're all interested in injury prevention, injury research, and injury treatment, one aspect or another. All the way from disparities and Antonio de Mayo's disparities, ORU, to the human genes that are being studied in CARTA, for example. We have clinical research, prevention, ethics, and interventions. The field of injury is one that is by its very nature transdisciplinary, whether it be behavior and aging, material science, health disparities, etc. The cost of chronic wounds, not the cost, the number of people with chronic wounds in this country alone exceeds that with Alzheimer's disease. Like Alzheimer's disease, it's got associated with psychosocial interactions, but unlike Alzheimer's disease, there's a significant amount of bias and prejudice related in part because it's a disease of this disenfranchised. It's associated with diabetes, with amputees, and often, tragically, the cancer survivor. But it's also a $25 billion problem in this country, and that's only chronic wounds. If you look at acute wounds, there are 40 million in-hospital surgeries that are done every year in this country, and each one of those is an acute wound. You can follow that up with 31 million that are, in fact, that are done in an outpatient basis. So this is a major problem that costs $12 billion for scars alone. So we have to ask the question, what can UCSD do to address this kind of problem? And what we would propose is a comprehensive wound research and prevention center that could really be a center for the entire West. There is none west of the Mississippi. If we were to put that together, then in fact we would have all of these natural interactions between all of these different groups, because this is a transdisciplinary problem. And the reason it doesn't have the highlights of Alzheimer's disease, for example, is because no field alone has taken ownership of wound healing. One of the ways to approach the issues with wound healing, I'm trying to get this into 7 minutes, is to use avatars. Now, to some of you, an avatar is going to be a representation of Vishnu, a small town in Iran, or this great science fiction book, or this heavy metal band. For the cases, for what I'm going to be talking about today, it's an avatar is really going to be represented by a model of a human disease. And classically, we would go from mouse to rat to pig to humans. And the success of these models has been absolutely extraordinary. No one will dispute it. But with the advent of HIV and AIDS, that changed everything, because the virus will not infect anything other than primates. So immunologists came up with the idea of, could we take what's the best out of the mouse and the best of what man provides and actually create a hybrid that would have the human immune system in mice. The process is pretty straightforward. You take CD34 positive precursor cells that you can isolate from blood. There's CD34 here. And now you have the donor. You take you engineer a mouse that can receive these cells. In this case, it's an NSG mouse that is so immunodeficient that, in fact, it has no immune system of its own. And when you give it these stem cells, then, in fact, you now have an animal model that has the human cells. What does this mean? Well, that means that we have a model that is neither a man nor a mouse, but, in fact, a model that is much more representative of the human system. In fact, if you look at mixed donors being the donors of the CD34 cells, you'll get a mixed population of mice. If you look at a single donor, then in fact you get replicates of that donor. So now all of a sudden, a whole field can open where we can study human biology in a way we couldn't study it before. And this is a specific example that Todd Costantini in our group did a couple of months ago. So here's a normal, healthy volunteer. You prepare cells, and you can create some 20 or 30 animals from this. If you injure this animal, you can see here, this is just the lung. You can see normally 78 human cells in that lung of this animal. If you injure this animal, you can see the increase. And if you, in this case, do a counter-inflammation experiment with vagus nerve stimulation, you can decrease the signal. The bottom line of this kind of study is that, in fact, we're able to reproduce the human reaction in an animal model. Now, this is something that has taken a huge amount of traction lately, as shown by these papers. And you can see that whether it be modeling malaria, for which you cannot model in any animals, and it's been a tremendous restriction to development, or sepsis, or diabetes, or arthritis, all of these can now be tested. But how does this apply to what we actually need to do in man? Well, the problem, and someone alluded to it earlier this morning, is that, in fact, if you look at similarities, these three can show up to the trauma bay, and one of them may be the one that doesn't leave. Nobody knows why. And that's where the omics technologies can come to play. But there are also major problems that particularly apply to wound healing as it reflects to age. The younger you are, the better you do. As it reflects to gender, women do better than men. And then finally, in the case of disparities, which I alluded to earlier, it's a major problem with wound healing technologies as they currently exist. But how does this then translate to a specific personalized? So you can take these cells and make mice that now have human immune systems, but we have the opportunity, if you've got tight collaborations with the clinical staff, of being able to take cells from a specific individual with a specific disease and actually make a true avatar of that disease. It's not a model of the disease, it is the disease itself, a disease in a cage, if you will. In this case, it's obesity, and Brian Eliseri in our group is actually looking at some in terms of keloid wound healing and things like that. Finally, our call really is to set up a transdisciplinary, comprehensive injury wound care response and prevention center, one that would be collaborating with the ORUs that are already established. And by the very nature of the complex biology for which no one has taken ownership, UCSD could be positioned to really play a major role in human health and care. Thank you. And it's the right thing to do.
That's a very interesting model. I was wondering if you have like a case example, perhaps, or a patient that you could describe this in the context of helping potentially.
Oh, absolutely. There are many kinds of examples. If you pick any disease in particular that's human-specific. So one of the things that we're interested in is the human-specific response to injury. Unknowns to most, perhaps, if you look at the response of humans to burn injury, the gene response is completely different, much longer, and it isn't reflected in current animal models. And there are also a population of genes which were originally thought that they were caused speciation, but in fact are turning out to play an important role in human-specific responses to disease. So diseases like keloids, for example, is another one.
Other questions or thoughts on this topic? Nice. That's great. Oh.
So given the number of people and the number of diseases that are there, are there any ethical things connected with mice, or it's not an issue at all?
Ethical things?
Yeah.
Absolutely. And in fact, I didn't have time to talk about it, but we have an ethics person from the CTRI working on our team for exactly that reason, because that's part of it. And part of it is, how do you engage the clinical team? Because you've got to be able to get these cells. How do you engage the team to purify the cells? How do you coordinate that with the animal quarters? How do you pull together all of the pieces to effectively be able to do the experiments you want to do? But this is the future. This is where all modeling of disease is going. Make no mistake about that. And that's because as all these omics technologies are coming out, it's becoming crystal clear that there are specific genes expressed in man that alter their responsiveness. And I can give you a good example of the acetylcholine receptor, where there is an isoform present only in man that is going to change the way man responds to vagal nerve stimulation, and that has importance for complementary medicine, it has importance for yoga, it has importance for everything that's parasympathetic. The virtue of that gene being present as a dominant negative inhibitor is going to change the way man responds and probably contributes to this variation that's seen between patients. Yes?
Human immune system. What about other models like the FRG liver, humanized liver model kind of thing?
Yeah, it's a question of being able to access tissues. So the process that we use is to be able to obtain peripheral blood. And the minute you use peripheral blood, then you have access to a wide range of potential donors. If you are looking to create, like a lot of the popular models now, as you say, using fetal liver, you have to get human fetal liver. And good luck with that. You can buy cells and make these models. And that, indeed, that is generation 0, where we are right now. But I'm not talking about generation 0. I'm talking about where this technology is going and where it's going to bring us. And where it's going to bring us is to an age where if we really want to find out what your cancer is behaving like, we will take your cancer, put it into the mouse that has your immune system, and we will know which drug we need to use under which conditions. And it won't be in a dish because we will know the issues related to the pharmacokinetics, we'll know the issues related to the pharmacy of the drug, and we'll be able to assess in vivo efficacy.
So does having, if your mouse has my immune system in it, does that mean you could put fragments of my organs into it and then use it to see how they'll respond in a living organism to various stimuli?
Darn straight. And in fact, the experiments I alluded to that we're currently doing and for which we just applied for center funding involve exactly that. It involves getting blood from donors 3 months before they come in for elective abdominoplasty. So after you get a lap banding, you lose weight, you gotta get rid of the skin. So we pulled in the surgeons, the plastic surgeons, the blood collectors, what are they called? Phlebotomists, thanks. I'm on the cloning side. And effectively what you do is you create a model in which you can now look at the interaction between of your immune system, your inflammatory system, and your skin. And this is particularly relevant for something like keloids, but which, is, and a lot of different scarring models. So you can go to that skin, scar that skin, take a look at it, and really develop therapeutics that hopefully will work in man. And that's the big problem.
I just have one burning question. What about spinal cord injury? Have you thought about any possible applications in neural-based tissue?
Probably not. But let me ask everyone here, how many here think that Christopher Reeve died of spinal cord injury? Probably you all do. He didn't. He died of a chronic wound. But you won't see that anywhere. And you won't see it because chronic wounds are basically a socially unacceptable. They're an ugly thing. And this is why I'm pushing for it, is that it's a field that's completely wide open for a group to take ownership of because of this transdisciplinary requirement that it has for treatment. But I won't grovel.
This is very exciting. I think this could be like a major leap, it seems like. That's fantastic. Thank you so much, Dr. Baird. Our
in this country, and that's only chronic wounds. If you look at acute wounds, there are 40 million in hospital surgeries that are done every year in this country, and each one of those is an acute wound. You can follow that up with 31 million that are, in fact, that are done in an outpatient basis. So this is a major problem that costs $12 billion for scars alone. So we have to ask the question, what can where UCSD might think of going in its strategic planning for human health and society. I really represent a team. We're actually a loosely grouped organization. We're informal, we're grassroots, and we're all interested in injury prevention, injury research, and injury treatment, one aspect or another. All the way from disparities and Antonio de Mayo's disparities, ORU, to the human genes that are being studied. The country alone exceeds that with Alzheimer's disease. Like Alzheimer's disease, it's got associated with psychosocial interactions, but unlike Alzheimer's disease, there's a significant amount of bias and prejudice related in part because it's a disease of this disenfranchised. It's associated with diabetes, with amputees, and often, tragically, the cancer survivor. But it's also a $25 billion problem in CARTA, for example. We have clinical research, prevention, ethics, and interventions. The field of injury is one that is by its very nature transdisciplinary, whether it be behavior and aging, material science, health disparities, etc. The cost of chronic wounds, not the cost, the number of people with chronic wounds in this country.
Our next presentation is Dr. Andrew Baird from Health Sciences and the Surgery Department on avatars of human disease, personalized modeling for personalized medicine. Very exciting.
Thanks. I'll try not to let you down. Thank you for waking everybody up, by the way. Don't start the clock yet. Thank you for the opportunity to share with you a vision for